Hi, everybody. We're going to start. So today we have with us Guillermo Fandos, who is working at the Universidad Complutense. And people are asking me, why do I know this guy? He did his PhD at the same time as my first PhD student in the museum. And he's already a faculty at the Complutense. So things are going right. So he did his PhD at the Universidad Complutense in 2017 with Tellería. Most of you know Tellería. He was working with models on migratory birds. He then did 2 postdocs, 2 years each. So he was 4 years altogether in Germany. First at the University of Berlin, Humboldt University, between 2018 and 2020, with Damaris Turrell, who's a young researcher who, along the way, he was doing a postdoc with this person. Is a he, right? Yeah. She got a permanent job, so it was a young group. So there he was working with models, so more transversal research, models based on processes. And then they moved together to Potsdam, where he continued working on the same things, but he got his first grant as PI there from the ADE. And then he came back with a Marie Curie, back with Tellería in 2022. And now Tellería is retired, and he has his own position as Profesor Ayudante at the Universidad Complutense. And he describes himself as a researcher, conservationist scientist, and he's interested in predictive ecology, and you're going to hear more. Thank you.
Oh, thank you very much, Marta, for the introduction and also for the invitation to be here. I'm super happy to be here. It's great to see familiar faces. And yeah, great. So today I'm going to talk about how we can improve biodiversity models for conservation in this global chain context. Okay, I think that many of you are familiar with this curve. Like we have this biodiversity crisis that we are losing 1 million of species of plants and animals are close to the extinction. And of course, models can be very useful for detecting these declines and see where and who is declining, but also for attributing biodiversity change. So, for example, what threats are behind these chains is habitat loss, is pollution, is hunting, overexploitation. And the models can be very useful for that. But the next step is like if we really want to anticipate these declines and to make like proactive conservation measures, is that we really need to forecast. And forecasting can really help us to be proactive. However, we still have a lot of uncertainty related to this forecast. And now we are in a perfect era where we have a lot of data, better computational models, where we can really reduce this uncertainty. Why is important this forecasting? We need to understand that we are in this loop. As a community of researchers, we try to provide guidelines for monitoring biodiversity at different scales, to detect biodiversity trends, and also to attribute these changes or these trends to different threats, of course, for forecasting. And these forecasting values are really important for policy. So we need these numbers. Policy need numbers. And this can be really helpful for management. And then it's a loop. So we have, again, like we can receive these numbers also as a community and then provide, again, guidelines for monitoring and, again, like feed this loop. So now during this talk, I'm going to talk about mainly the research line that I have, like how is biodiversity distributed in space and time, and how does it respond to environmental change. And I will focus on these 3 key steps, like, first of all, improving the biodiversity monitoring and management, then how important is to understand the relevant process, ecological process at different scales, and finally, how we can improve biodiversity model by calibration, sensitivity analysis, and very important, like how we really need to understand this uncertainty and how we communicate and how we can reduce in our models. I think that all of you are familiar with GBIF, and here we can see a geobond animation where we can see the green lights is GBIF occurrence, and very clear you can see like we have thousands and thousands of millions of data. But of course, this data comes with a lot of biases. So we have biases in space. As you can see in the previous animation, this data is not homogeneous distributed in space. We have biases in time, like we have more data now than 20 years or 10 years ago, even 5 years ago, and biases in taxonomy. Of course, we have some species that are more focused. I mean, we have more data for some species, and for others, we don't have any data. And of course, we need also method for analyzing all this. So we are entering in this big data era. So we have this spatial bias. That this is an example of a lot of studies that I did during my PhD. Here in the right, you can see green recoveries from wintering birds, and in the left, you can see human footprint. That is a level of human influence. And as you can see, it's like a really good correlation between both. So if we want to use this data to infer where the birds are wintering or where the birds are distributing, we really need to account for this bias to be sure that we are predicting where the birds are and know where the humans are looking for birds. This is another example of this taxonomic bias. So you can see here all the priority species, but here you can see like only a small group of these species are used as a species indicator. So that means that we don't know a lot for a lot of species. And to use this data, I will present you like how important it is to disentangle the observation process and to correct for imperfect detection. So here is the pattern distribution, the occupancy pattern distribution, where you can see, for example, the deers in the space. And of course, we have the ecological pattern, so the real pattern that we want to study, like why the species is there, where the species are, and everything. And this is the real ecological pattern. But we are not able to see that because we always have this observation pattern. And if we really want to understand where the species are, why they are there, or when they are moving, or everything, we really need to disentangle these two patterns, this observation pattern and ecological pattern. And I will present you some examples of why this is important or how we can do that. So for example, we have the problem of the preferential sampling. Here in the left, you can see random, so designating sampling locations randomly, but this is not always the case. I mean, sometimes we have the problem that we have abundance bias. The people, researchers, or whoever that going to sample this site prefer sites that there are more birds or more butterflies or more animals or more plants, or richness bias. So sometimes we go to sites that we know that there is a lot of species to study and to have more information, to save money of going to different places. And not only the citizen science have this bias. We need to understand that this is not black and white. We have like a really great distribution of these biases across different data sets. And even academic research data sets can have some preferential sampling. And this is, for example, one of the cases, like here in this study, we try to understand what is the occupancy and reproduction dynamics of a peripheral black stork population. And this is quite important because here in Spain is vulnerable. So we really need, it's important to understand these dynamics to really see how it's going with this animal and how it's going with the populations and what are what threats are behind these dynamics. We were using an amazing data set from more than 20 years of these nest dynamics. But as soon as we look deeper on this data set, we saw that we have preferential sampling. So we have the problem that the site quality, so for example, nests that are more successful, more chicks every year, they are more probable that they have been visited, no? Like the people going to visit this successful nest. And if we are not taking into account this preferential sampling, we can really overestimate the population. So, for example, here you can see that the blue line, that is the important that I want that you look, is where the model that we take into account the dynamic, but also the preferential sampling. And you can see like how we are with the rest of the models overestimating the population, in some cases like more than 20%. And for this kind of a species and in this conservation context and framework, this could be very, very important. I mean, like it's the meaning like we make this species threatened, vulnerable, or even non-threatened. So these numbers and these bias is quite important to understand. Now, as another example that I am currently working on, I am using structural surveys with distance sampling protocols, and I am using M-mixture models to estimate population relative abundance across time. For different species, we have this kind of graphs for different areas, and this is super cool because we have this probabilistic sampling design that help us to disentangle in this observation process and ecological pattern. But we have some limitations. Of course, we need a lot of resources to have this sampling design, and this is not always possible. And we have limitation in terms of spatiotemporal extent. Of course, even it's more difficult to have these long-term studies across time. On the other hand, we have citizen science data that we can also use these models to have relative population abundance and see how it's changing in the different areas. But the good thing is like we can cover really large spatiotemporal extents, but we don't have sampling design, so we are not able to disentangle in this observation process from the ecological pattern, and therefore we have an inaccurate understanding of the ecological process behind these dynamics. What I am proposing, I mean, many people are working on that, is like we integrate them in a model. So here you have the hierarchical model, where we have the standardized surveys and the citizen science, and we can have the observation process for each of the datasets. And what we really want is that the population dynamics. And both datasets have information about these population dynamics. And this can help us to have like more accurate sense or accurate view of like how this relative population abundance is changing through time. But more importantly, we can upscale these dynamics because we will have this information for larger areas and larger times. So we have a scaling parameter. So the parameter, the standardized surveys is going to give us the parameter. And what we are going to do is using this scaling parameter in the citizen science data. We are going to improve our precision parameters. We can do it at broad spatiotemporal extents. But of course, we have more model complexity. And this is something that we really need to deal. We need to validate. We really need to understand that we need to make sensitivity analysis. We need to make sure that we understand what is behind the model. But the really nice thing, and what I really want to introduce you, is like we can really improve our forecasting predictions. We are not really improving these forecasting predictions in terms like giving very different signals, but what we are doing is really reducing the uncertainty. And this is crucial to know, like, to have a better idea of what scenario we will find in the future. Now I want to speak about, like, I have been speaking a lot about data. Now I want to speak about, like, modeling. No? I mean, I think that you are familiar with this hexagon. Urban and colleagues, Mark Urban and colleagues, define these 6 key mechanisms that we really need to introduce in our models to really understand biodiversity response to global change. We have adaptation, environment, physiology, demography, dispersal, and species interactions. And we are not really doing that. I mean, only 25% of the studies study or introduce at least one mechanism, and usually the rest of the studies is like 2 or 3, but we are really missing a lot of information here. And another important step is like we really need to understand that biodiversity have this hierarchy or the different scales important, no? And we really need to understand the different ecological processes that play a role at different scales. So we have processes that play a role at the individual or population level, and another process that can play a role at community or ecosystem level. And the problem is like all of them is quite connected, and we really need to understand that we need to monitor biodiversity, but also make cross-scale model assessment. I'm going to speak about a bit of adaptation, not in terms of genetic, but more in terms of I'm working out with animals, and they have this behavior plasticity and movement. I think that maybe some of you, like me, are fascinated about the animal movement. Here you can see a Max Planck a video about like how different individuals that are all of these lines are moving across the space, looking like in these seasonal chains, and it's quite impressive how the movement of this biomass across the globe. And the movement can really help us to make this cross-scale assessment. So here is an example. So now we are going to focus on the white stork, and you can see some individuals moving across the year between the breeding grounds in Europe and the wintering grounds in the Iberian Peninsula and Africa. And all these birds are 64 white storks with GPS. And one of our questions was like, okay, so we can see that like there is evidence that the species and the populations track the needs across the year. So look for the similar environmental conditions during the different seasons. But we don't know where is coming this pattern. No, it's coming. The individuals are doing the same. The individuals are looking for the same environmental conditions. They are not looking for that. So we have an amazing data set to answer this question. And we were surprised about like the pattern that the species and the populations are tracking the climate needs. We cannot see that at the individual level. So the individuals are more worried about what is the wind and the weather that is doing in this current situation, but they are not worried about the climate in the last 30 years. No? So this can give us an insight about how these processes are more complicated at the different scales. So no, we have emergent properties at the population, at the species level, that the mechanisms of the individuals, they are not following that. So we need this cross-scale assessment to really understand how the species can respond to global change. Another example, this is with a collaboration with Rafa Barrientos, that we try to measure the plasticity of the mountain lions to like how they respond to light pollution. And we saw like we can have like an average population response of negative, of course, so they try to be far from light. But what is very important also to take into account like how they are responding is like you can see there are some individuals that really don't like the light, and there are some individuals that are more tolerant. So sometimes we are really neglecting all this variation when we are making our predictions to the future. So here, again, we made a review to see how we are really applying these models or the different models in conservation and restoration. Here you can see the different hierarchical levels. And as I told you, like we have a lot of models here in population and species, but we have really a lack of understanding of what is happening, for example, at the gene level or in the ecosystem. But very interesting, like we divide in static and dynamic models, and almost 80% of the studies apply a static approach. So we are neglecting this dynamics in biodiversity. And this static approach, this correlative species, I think that you are also very familiar with that. Usually we need some kind of presence data, abundance, and we combine with different environmental variables. We can take into account bias sampling, and with different algorithms, we can predict the suitability habitat for different species. And they are very, very useful. They have like a lot of advantage. And for example, we use these models to try to predict how the winter distribution of different migratory species is going to change in the future with the climate change. And here, in another examples, with Alfredo Romero from Humboldt University, we try to use these models to understand how the biodiversity, or the mammal biodiversity, change like from the 80s until now, and how the different biodiversity facets, not only the richness of the species, but also the phylogenetic diversity and the taxonomic diversity, change for different threats, that habitat loss, hunting, and the combination of both. And we can see that in almost all places we lost biodiversity in the different facets. But what we are missing? So if we really want to predict species range dynamics, as I say, like we usually apply this static approach that can be very useful, can really give us a lot of information, but we are missing these dynamics. So if we apply this, I am suggesting that we really need to move forward more in a movie approach, where we really can understand some processes that are behind these dynamics, like, for example, colonization and extinction dynamics or dispersal. So, for example, in a specific case, like where we want to see like where and when large carnivores will likely expand into human-dominated landscape, and try to highlight where proactive conservation measures can really help us to reduce this human-wildlife conflict, we introduce dispersal and make these models dynamics to really see how this colonization and extinction dynamics can be really affected by anthropogenic disturbance. So here, for example, you can see the static approach, where we have in dark red where the species actually are, and in light red where is the suitable habitat. And our question is like, okay, how the anthropogenic barriers will affect the colonization of this suitable habitat if it is happening. So we make these models, including dispersal, and we can see here in the right, for the Iberian lynx and the bear, how the high scenario of high anthropogenic barrier can give us really different places of colonization than a scenario of low barrier. So we can really identify those places that where a proactive conservation measures can really impact in this colonization process of the large carnivores in the Iberian Peninsula. So I hope that I convince you of like, okay, how cool is going to this dynamic approach, but why we are not doing that, is worth it doing that. So here we evaluate, we compare with a data set from Swiss birds, so Swiss monitoring data set. We compare this dynamic and static approach and try to see if we are really improving our spatial prediction, our temporal prediction, if we are using dynamic or static approaches. So you can see here different models where we take into account the detection, don't take into account the detection. So we play a bit to know what we are missing or what we are really understanding if we are using into a dynamic. Because we really need to understand that dynamic approach, of course, they are super cool, but you need a lot of data for that. And here is an example with the Eurasian skylark, where you can see where we evaluate the models, and you can see here the spatial evaluation and the temporal evaluation. And the first answer is like, we don't have a best model always. So that means that depends a lot of the question that we have, we really need to use one model or the other. For example, for the spatial predictions, we saw that the static approach, the booster regression trees, have the best evaluation. So we have the best spatial predictions estimates with this static approach. But for example, for the temporal, to detect these trends across time, we have this dynamic have the best evaluation. So depending on what we want, depending on the data that we have, maybe it's worth it or not to move into a dynamic approach. So again, like why we are not using these process-based models, similar thing. We have a lot of data limitations, computational challenges, and we really need additional expertise to move forward with that. So one thing that, together with Damaris, we try to understand is like, okay, if we really want to introduce, for example, dispersal as a key process to include in these models to understand how the movement of the individuals scale up to population patterns with the gene flow and everything. Why we are not using that? It's so important that our question was like, what is happening? And when we try to look deeper of like, we have dispersal data for several species, we don't have this data. We identified this amazing paper from Emmanuel Paradis in 1998 that estimates dispersal for 75 species, but it was only in UK. And we really need to understand that this data set that is super good, but until I think that 1 year, 2 years, it was the only empirical dispersal data set for several species in a standardized way. So we decided, okay, we really need to move forward with that, and maybe we can apply this method now for more data. So we have now the urine database that is a volunteer-based monitoring database where we have all these rim and recaptors, and it's managed and coordinated by each country. And we say, okay, very good, we can use that to estimate dispersal for more species. And at the end, like we realized that why the people were not doing that because it was like very, very challenging. We have the data set, but we have a lot of biases. So we have the spatial bias of the recovery. We have the migratory bias, so it's crucial to identify these migratory individuals to exclude from the data set, because if not, we are going to overestimate dispersal. We have a scheme report heterogeneity, so the different schemes in Europe try to report the data in different ways. So it was very, very challenging, but at the end we were able to estimate dispersal kernel applying different functions, and with the dispersal kernel we provide more holistic view of this dispersal pattern. And we did that for 274 species of European bird species. And with that, we hope that this can be very useful for different things. One thing, for example, that can be very useful is to understand more the dispersal patterns in the European birds. So for example, here you can see our dispersal estimates for the breeding, natal, and average dispersal. And here in the right, you can see like how this dispersal covariate or is correlated with different traits for the different species. So we can see that body mass and life history and diet seems to correlate quite well with dispersal. So larger species disperse more, and carnivore species disperse more. So what makes sense, and we really understand what traits can really covariate with dispersal. And the next question that we had is like, okay, if we know what traits covariate with dispersal, maybe we are able to predict dispersal using a trait-based approach, no? And for many species, maybe we don't have this information, we don't have dispersal, but maybe we have information from the different traits, and we can use these traits to predict dispersal. And what we found is like, actually using all the traits together, we really have really bad predictions. I mean, there are many things behind there, many processes behind these covariations that introducing all the traits together, we have really bad predictions. And our best predictions, especially between orders, so when we are doing predictions in a really different from a phylogenetic point of view species, single traits like body mass or hand-wing index or different single traits are really improving our dispersal predictions. Even that, we are not above 50% of this dispersal prediction accuracy. But the future perspective that I want to give you, like as soon as we have all this data available, we can really move into process-based models. And these process-based models, this work was developed by Anne-Kathleen Malchow, and she developed this rain shifter R package. That rain shifter is super helpful because it's calibrate individual-based models, but it's modular, and we can really include, for example, dispersal, demography, genetics, environment, and we can include all that in the model to really understand how this different process is affecting to the range dynamics, and it's also especially explicit. So really helpful for understand and improving our biodiversity dynamics. And the good thing here is we can really integrate data from different sources. So we can have expert opinion, bibliographic data if we don't have this empirical data. Then we can include monitoring surveys, abundance, trends, present, absence, and also ecological theory about, for example, when the species is reproducing, when the species is dying, a lot of the demographic parameters and ecological theory process. And also use this Bayesian calibration. And I really want to stop here a bit because how important can be that to understand our uncertainty in the data. For example, if we have, we can generate prior, for example, with data that we know, also from the bibliography or from different data set, we can generate this prior knowledge, but we also have a likelihood. And with that, we can multiply this prior likelihood, and we will have these posterior estimates. And this is very important because we will have a sense of the confidence of the data that we have. So if we are more confident or not, and this will be a cascade effect into our predictions. So we will have our predictions, and we will have our outcomes. Our outcomes will be also these parameter estimates and this posterior distribution. But then with the predictions, we can compare alternative scenarios, project to different times, different areas, and also have these credibility measures. An important thing that also Anne implement here is this validation. So we have really strong protocols for validate static and species distribution models, but these process-based models are not always validated. And here we use a spatial cross-validation to validate that. So we can really have a sensitivity analysis of how well our model is working. And we implement that for the red kite in Switzerland that previously, 2 decades ago, there was really, really low population abundance because persecution and many predators in Europe. And we can see here the spatial predictions to now and on to the future. And here we can see the temporal prediction. And in red, you can see the observed values. And in blue is the model predictions. And we can see that when we have observed values, it's the model is working quite well. And with the predictors in the future, we can more or less assess that the population is going to be stable in 20, 40 years. So with all that, the take-home message that they want, that you can get from this talk is we really need model biodiversity assessment for proactive conservation measures. We really need to develop a monitoring framework across ecological scales. And I know that GeoBond is doing a super good job here, but we really need to understand how important it is to monitor from genes to ecosystem. We need to improve the calibration and the validation of the dynamic models. So we really need a statistical toolbox to develop these models in more accurate and in more robust way. And very important, the analysis and the communication of the associated biodiversity data and modeling uncertainties. We really need to understand how important it is that we communicate that to really understand how we can implement these values in policy. So, of course, this is teamwork. Here is part of the work that from these collaborators. And thank you for all of you for listening.
Thank you, Guille, for this super interesting talk. So now we open time for questions.
Thank you very much for the presentation. It was really interesting. And I just have a question about the niche of the species. So when you part from some data, are you predicting from the niche that this data is representing, or are your models able to consider some shifts in the niche that we know from the data that we have right now, that it doesn't really reflect the entire niche of the species?
Yeah. So do you mean like with these process-based models?
Yes, like in the last example with this hawk, I think.
With a red kite?
Yes.
Yeah. Yeah.
So you see, like the prediction is underestimating the actual next steps of the species that can colonize other environments.
Yeah.
That may not reflect the actual niche that the species is living in. I don't know if I make myself.
Yeah, so you mean like if it's changing the needs of the species when we are, yeah, of course this could be a limitation of the data. Like we don't know like if it's changing totally the environmental condition or we have this bias. Yeah, this is a limitation. But the cool thing of these models is like we reduce to the individual decisions. And this can give us a lot of flexibility in the ecological process that are behind that. And if we are incorporating all the data together, not only the present and absence, but also like if we are really making the point of, okay, the dispersal is really affecting to that, maybe we are improving that. But of course, it's a limitation that we are always going to have. If the species is totally changing the needs, and for example, with invasive species and so on, is a challenge, but we have the limitation of the data.
Okay, thanks.
Yeah, thanks, Guillermo, for a really interesting talk. I really love that you focus on uncertainty, which is the main issue here. And I wonder whether, because you already said that, okay, you can only explain about half of the variation with dispersal. Okay, that's a lot.
Yeah.
That's really a lot. And then you come here to range shifter. So are you trying to embed some dispersal modules in range shifter that you can use to include priors about dispersal and then later on improve the models, etc.?
Yeah, very good question. Actually, we are working on that now. I mean, like, as you say, like what we want is, okay, we have this dispersal estimate, and of course we are using this estimate in these kind of models to infer demographic response, to infer everything. But also we are trying to do this inverse step and try to see if from this likelihood and this prior, like playing with the prior knowledge, we like having a sensitivity analysis of how the different decisions about dispersal or how our information about the different process can affect to the predictions. And we are working on that with the dispersal because we know the true, we know the truth. Like the good thing is like we are providing dispersal kernels, so this can give you like more like the distribution of distance that can really affect, and you can focus on the long distance dispersal, you can focus on the median or the short distance, and we are playing with that to see how change the predictions and everything. So the important thing here is like with range shifter, we can really go down to the individual level, and with the Bayesian calibration, we can really have a cascading effect of this, how sure we are about our data. And preliminary results seems that is quite robust as soon as we are playing this sensitivity analysis. And with the knowledge, of course, we will have better or worse predictions, but at the end, with the uncertain, we are very clear communicating how sure we are about our predictions.
Yeah, now that you talk about communication, because that was my second question, have you tried to communicate, because communicating uncertainty is really difficult, and we've seen it with all the global change impacts we're having. So have you thought about the way of communicating this, because this translates into spatial uncertainty about the models, but also into uncertainty about how far the ranges will shift or how strong the impacts of different scenarios will happen. Have you thought about that?
Yeah, I think that it's a very actual question, and I think that it's more an answer of us as society. We, policymakers, society is pretending that we really communicate really precise results. Okay, this is happening, this is not. And as soon as you are communicating, okay, we don't know, we are not sure about that is the real thing, that we are not sure about many things, and we are communicating with this uncertain, I think that sometimes it's very like a gap of communication there. And I think that the first step is try to communicate in a very realistic way, in a very explicit way, this uncertain, and to the society and to the policymakers and to the managers.
Yeah, but have you tried to map it with this? I'm putting it into my place, but also have you tried to map this uncertainty?
Yes,
because maps are really powerful.
Sorry, I didn't answer. Yeah. We are mapping also this uncertainty. It's more like Anne just published, is in Bioarchive, a new paper about how communicating this uncertainty, doing this sensitivity analysis. So it's still in Bioarchive, but I think that soon it's going to be published, and is really focusing with Florian Hartig in this communication of these uncertainties and how important is that.
Yeah, thank you, Guillermo. Thanks.
Thank you. That was just a really, really wide-ranging talk on so many things that are really interesting. So thank you. Going right back to the beginning and thinking about sampling bias and different recorder effort. So you were thinking about that with GBIF data and with dedicated scientific surveys. Do you think about using social media data at all?
Yes. I mean, like it can be like another data set. The nice, I mean, of course, when we say citizen science, we have different levels of citizen science data set. No? We have some data set that have more information, they have like closed protocols, and others like social media can be like eventual or opportunistic insights. And I think that even that can really give us some information. So what I am trying to communicate and trying to move forward in the field is like everything has some advantage and disadvantage, and we really need to evaluate. But as soon as we are controlling this observation process, and we need to think how we can control the observation process of the social media. But as soon as we can do that, it's very nice to introduce in these models. So the nice thing of this is these models are super flexible. And I think that the challenge in there is like actually understanding what is the model doing behind. I think that here the simulations play a role, but I have been playing a role with citizen science that I reduce in some places, I reduce temporal, and see how robust are our models. And the good thing is, like, as soon as you integrate with standardized surveys, these scaling parameters really help you that the citizen science is not giving you really a lot of noise. So you are controlling that.
Do you think that would work if your social media data if people from social media tended to note species in really very different locations to the more, if the data were complementary but a bit different?
So you mean like they are in different spaces? I didn't try that. I mean, you need to make the strong assumption that the process, what is affecting the process is the same in the different places. Could be, but it's a strong assumption. But yeah, thanks a lot for the question.
Thank you, Guille.
Hi, Anna.
Super nice talk. Following up on Regan's question, so I mean, I understand that when you have the citizen science data and you couple it with standardized surveys, then you can really, gain from both type of surveys, let's put it like that, and have a better model. But what happens when you don't have those standardized surveys? What shall we do with data that is collected in a citizen science approach, and how can we make the best out of this data to provide good ecological inference? And I will talk more about this with you later.
Okay.
So.
Yeah, no, actually, before these models, I mean, I didn't put all the examples because I didn't have time. But before this model, I was playing a lot along with the citizen science. And as I said, like, there is different levels of citizen science. And we have, for example, EBER data that can give us a lot of information about the sampling effort. And this is crucial to really take into account and control this observation process. And at the end, we have a really robust estimate. The problem is, like, if we are not able to control this sampling effort, it starts to be a very specific case.
I'm going to speak very loudly. But what about when you have more or less... information about the sampling effort, but then, of course, the design of the study is really lacking. Like, I mean, you have a random distribution of observation points. Let's call it camera traps.
Yeah.
For example. Very specific.
The nice thing is with camera traps, and if you have sampling effort, you can do magic. Yeah, I mean, like, you can really get robust estimates. Of course, you will have more uncertain, and you have like less robust estimates, but at least you can be sure that what is you are getting there is taking into account this sampling protocol. Of course, in specific cases, there are some cases that no, but with camera traps, actually, these hierarchical models are working quite well. Actually, there are many people developing, so it's a good example. This social media will be more challenging.
Do we have any additional question? No? So we'll leave the rest of the questions for the kiosco. Then thank you again.
I want to say only one thing, that if you are interested or you are doing similar things to that, together with Maria Paniu, we are organizing a thematic symposium in the Spanish Ecological Conference in Pontevedra next year. So I'm really pleased to meet your abstract, and we can make a better community of this forecasting biodiversity. So yeah, you are more than welcome to see there.